For you to sit down. Okay, so my name is Sagiv Shifman from the Hebrew University of Jerusalem. I'm a biologist, yes, a real biologist. So statistics and programming are just a hobby. So my main interest is to understand the underlying biology of disorders like autism and other neurodevelopmental disorders. Okay, so this is what I'm going to talk today. And we heard quite a lot of talks about solutions, about computational solutions, but I really want to talk about problems, not solutions. And I know that here in the US you don't have problems. You have only challenges.
And solutions.
Yeah, but I'm coming from Israel, and so we do have a lot of problems. Okay. So I'm going to talk about all the problems that we have, some of our naive attempts to study autism. I want to start by talking about differences between expectations and reality. So many of the biologists have all kind of expectations, or every time we have a new technology, we think this will solve everything. And so this graph, which is a very accurate, yeah, I've drawn it myself, and show you that the expectation every time there is an introduction of a new technology to the field, and people think this new technology will solve everything when we're going to understand all those complex disorders. But in reality, we have disappointment, and after some time, you face reality, and you see that those methods advance the field a little bit, but still we're very far from really understanding those disorders. And I just want to give you just one example of this disappointment that was published in Cell just now. So this is just an examples of new technology like microarrays with and then new sequencing technology, and the GWAS with the looking at common variation, thinking that this will solve all the problems with understanding the genetics of complex traits. Then the genome sequencing technologies and the looking at rare variants. And as I said, I want to show you just an example of disappointment. This is a paper that had a lot of, I would say, reactions for human geneticists from Jonathan Pritchard lab, published just now in Cell. And the problem that they've emphasized in this paper is that many complex traits are driven by enormously large number of variants of small effects, potentially implicating most regulatory variants that are active in a disease-relevant tissue. So the main problem is that the whole genome is associated with disease. So if everything is associated with the disease, it's like saying nothing is associated, because we don't have pathways, and we have just every variant that will affect the tissue relevant to the disease will be associated with the disease. So this is a disappointment, but you have already a solution. So this is the solution in the paper. The next most promising step is to hunt for lower frequency variants of larger effect. And I want to show you in this talk that this has already been studied, especially for neurodevelopmental disorders. And so we can face the next disappointed already now. Okay? Okay. So in my lab, we do all kind of things. As I said, I'm a biologist. So when we talk about models, we normally talk about those kind of models, okay? Cellular models, mouse model. I'm not going to talk about them, although we have very exciting results, and I'm going to focus on our naive attempt to understand autism in a kind of a more system way, trying to link genetic variation and the functions or the activity of those genes in the brain. Okay? So I'm talking about neurodevelopmental disorders, but which are very prevalent in the population, 5 to 15%, many different disorders, deficits in cognitive, language, social skills, sometimes also motor skills, also more prevalent in boys than girls. But I'm going to focus, as I said, on autism spectrum disorders. Okay? But many of the things that I'm going to tell you are relevant to other neurodevelopmental disorders. And from genetic point of view, there are very different variants that contribute to those disorders. There are de novo mutations, new spontaneous mutations. There are rare inherited mutations. And I'm talking about variants that are very, very rare in the population, but are inherited in families with autism. And of course, there are the common variations with large frequency in the population, relatively, those alleles that are being studied by genome-wide association studies. Most of the genetics, based on studies that have been done in the last few years, are attributed to common variations. Okay, here it is. This is from my not very recent review in Neuron by Dan Geschwin and colleagues. And in yellow, these are the, doesn't work, no. In yellow, these are the amount that they thought at that same moment that is contributes to autism. So based on this kind of pie chart, we should focus on common variation in autism. But in fact, there was very limited success in genome-wide association studies until now for autism. This is just an example from 2009, which looks like there is maybe one single region that is associated with autism, but this was not replicated by other studies. There are larger studies now that have been published for autism, something like 16,000 cases, and the picture doesn't look very different. Okay, so until now for autism, with the limited sample sizes that were used, we don't have very good information about common variation. What I mean is that we actually don't have genes that we can link to autism, but we could study the polygenic score or the contribution of multiple variants together and show that common variants are important for autism. But again, we don't have any specific genes. Same with rare inherited variants. We have no knowledge whatsoever about them, almost no knowledge, I would say, about rare variants which are inherited in families of autism. As already we heard here in this course by Jay Hoon yesterday, it's very hard to do an association with rare variants and which are inherited in general because there are many of them in the genome. The statistical power is very limited. There are all kinds of statistical methods that aggregate those variants together, but still we don't have very good results or studies that show the contribution of rare inherited variants for autism. So everything we know about autism actually comes from studies based on de novo mutations. Okay? So just to remind you, when we talk about de novo mutations, we talk about a family, okay? We talk about mutations that occurs in the sperm or in the egg, transmitted. We're looking at those de novo mutations that are transmitted to the affected child, and compare it to the healthy sibling. Now, while you're laughing here, I want to again emphasize that if you think Trump is a problem, he is not. He is just a challenge. Okay, you're in the U.S. Okay, what? Result. Computational solution, you mean? Okay. So de novo mutations. Again, I'm talking about mutations that we see in the kids, especially those with the disorder, and we don't see them in the parents. And again, those occur in the generation of the sperm or the egg. Okay, so most of our genetic information or specific genes that we know linked to autism come from studies of de novo mutations only. Okay, the story about de novo mutations in autism starts around 10 years ago. This is just an example of one of the first studies in science from the lab of Wigler. First author here is Jonathan Seba, and they've showed that when they look at copy number variation and de novo copy number variation, were much more frequent in simplex families versus control. Simplex families are families that have just one child with autism. Okay, so as you can see, the numbers here it's almost like 7 or 10 times more frequent to see those large de novo copy number variations relative to the controls, and they saw a non-significant increase in those de novo copy number variations in multiplex families where you have multiple cases, multiple siblings in the family with autism. Okay? Yes?
Do you by any chance remember the absolute values, like how many
the absolute
copy number variations are you supported?
So those kind of copy number variations, de novo large copy number variation, you normally see, if you see them, you see only one in an individual. Okay? Normally. Sometimes you can see more than one, but not much more than one. Okay?
If you have two brothers without it, then they generally don't.
In just a normal population, you're going to see something like 1%
But
of
it's
cases.
interesting
And those 1% of cases will normally have one such deletion or a big deletion or big duplication. What? Sorry?
How large?
How large? This could be a few hundred KB, few megabase. Depends. Okay? At that time, the technology was microarrays. Those microarrays, you can use them also, of course, to identify those copy number variations. So people were very happy with those results, but people from the Wigler lab were not happy enough to say that copy number variation, just de novo copy number variation, contribute to simplex families, but they actually designed a model or claim that the copy number or those de novo mutations could explain both simplex and multiplex families. And this is the paper in 2007 that they published. They called it, look at the title, A Unified Genetic Theory for Sporadic and Inherited Autism. And they say that if you look at a model where de novo mutations are dominantly acting and they have reduced penetrance in females, you can explain almost everything. And the model, just without numbers and equations, is like this. You have a de novo mutation. If it occurs in a boy, very highly likely it will develop autism. If you have autism, the chances that you're going to have a family and kids are very, very low. So this will be the end of it. If the de novo mutation happens in a girl, okay, because of low penetrance, there is a high chance that she will not develop autism, but she will transmit this mutation to her kids, and you're going to get a high-risk family with something like 50% of the boys in this family will have autism. And they took lots of families and showed that this kind of model could explain the inheritance in those families. Okay?
So the data that you're trying to explain was the table that you showed?
No. So they've took families with autism, okay, and just showed that they can explain the way the inheritance happens. So they can show that there are high-risk families, where in those high-risk families, in the boys, it was like 50% having autism or not, which goes with the option that there is one large mutation with a large effect, which is dominant, okay, and other families which are sporadic, okay. So it's not based on the genetic data; it's based on the inheritance in families, okay. So yeah, copy numberation, de novo mutation could explain everything, okay. We're very happy about it. But I promised you that I'm going to talk about problems, not about solutions. So in the field, yeah, people move to studying rare variants. This is, for example, were few papers that were published in Neurons talking about copy number variation and autism. And I want to show you now the genes that were published in just one of those studies, okay? So yeah, this is the list of genes in de novo copy number variations in just one of those studies, okay? You see many genes here, and this is because, first of all, those copy number variations are very large, includes many genes, and almost in each individual with autism, you have a different copy number variation in a different place in the genome, almost in any of them. So we have a lot of problems. As I said, each copy number variation has many genes, so it's a bit of a problem like we have in cancer. That's why I've called it that we need to distinguish between a driver and a passenger. It's a bit similar because in those big, large copy number variations, we assume that there is maybe one or two genes that contribute to the disorders, but the other genes in the deletion or duplication are just there as a passenger just because they were close to a causative gene. Okay? So we actually don't have very good methods. Yes?
Is the extent of the copy number piece of it, like how long it is or how much of it, related at all to the phenotypic, like where they are on the spectrum? So the degree of disease related to.
So the question is the association between the size of the copy number variation and the phenotype? Yes. So the answer is yes. The association is stronger for larger copy number variation that maybe hint to the possibility that there are more than one gene in this region, but still there could be a lot of passengers in. So we see a lot of effort in cancer to try and solve this problem, but I didn't see a very good solution for copy number variations in autism. And as I said, there is another problem is that in each one of the individuals that we look, we see another copy number variation, almost in each one of them. There are a few that are that you can see that are the same.
I'm sure people have tried to see if these different variants share something. Are there some genes that are
So
recurrent?
exactly. So this is the question I'm asking here, whether we can find any shared processes or pathways of those genes. This is one of the questions that we were trying to address. And this is also finding those pathways maybe could also help us to identify which ones are the drivers and which one are the passengers.
Location.
Again?
Location.
The location?
Is there a pattern in terms of where these things are?
Okay, so the question is whether there is a pattern or whether where those copy number variation occur. Okay, so of course they don't occur uniformly across the genome. There are regions that are more susceptible in the genome for breaks and for this type of copy number variations. So for example, fragile sites will be associated with those deletions and duplications. Recombination could be associated with this.
This is kind of a
But-
neutral mechanism,
Yeah.
meaning that it would happen for any, not necessarily the ones associated with autism.
Yeah.
But there is no evidence that the ones associated with autism tend to be, say, on one
So-
chromosome in particular.
No,
or-
it's not just in one chromosome, but you're going to find many studies that people were attempting or suggesting that they can see some processes or pathways that are enriched for the genes in copy number variation. Okay? So, as I said, our attempt was for several years to find some processes or some convergence of those genes, not just copy number variations, but in general, to see whether there is anything that is shared among those genes. Okay. One such an effort that we published in 2012 was done by Eyal Ben David. He is currently a postdoc here in UCLA. And his attempt was to see whether we can see a convergence, whether we can see that rare variants and common variants affect the same genes or very, very different type of genes. So one of the problems is how you define a group of genes that are similar to each other. There are very different ways. And we decided to use gene expression as a way to divide the genes in the genome into groups of related genes. At the same time, the Allen Brain Atlas published a very big project where they've used microarrays to profile gene expression from different regions of the brain. Okay, they have hundreds of different regions of the brain. They've measured gene expression across the whole genome for each one of those genes, and we used those measurements to generate a gene co-expression network analysis. As I wrote here, we use this approach to divide the genes into groups, to modules, and then to test the enrichment of mutations, rare mutations, and also common variants in each one of those groups. So we used an approach that was developed here in UCLA by Steve Orvas. It's called, as I wrote here, WGCNA, Weighted Gene Co-expression Network Analysis. I'm not going to go into the details how you calculate it, but I just want to give you, in a very short way, the principle of this approach. I didn't say that once you divide those genes into groups, you can always go back and take those groups of genes back to the brain and see where they are expressed and try and connect the enrichment to brain regions. Okay, so
in human brains you were looking at where they're
yes,
expressed in data
this is
base?
human brain. As I said, this human brain was dissected into hundreds of different regions. Each region was profiled for gene expression. Okay, so we had hundreds of those measurements. Just two brains, two healthy human brains. Okay, but many, many different regions.
Autopsies from
People who died, of course, because nobody will give his brain when they are alive.
I was asking.
I mean, I would not give it, but
All right.
okay. So just to give you, I mean, you can read about it or see if you're interested, but just to give you the main principle of this approach is, First of all, it's based on the correlation between the genes. Okay, you have a big correlation matrix. In the WGCNA approach, they use a power between 6 and 12, which means that it's a weighted network. You don't have a threshold saying, for example, if my correlation is bigger than 80%, the genes are connected, and if it's below, they're not connected. It's kind of a soft threshold. And the argument is that by using a power on the correlation, you get a scale-free distribution. And actually, in the program, they use a scale-free distribution to choose the right power, okay, the beta. Okay, so this is one principle that they use. Another thing is that they use what they call a topological overlap dissimilarity, which means that to see how to measure the distance or the similarity between genes, you don't use only the correlation between the two genes, but you look also at the neighbors. Okay? This is another principle in this analysis. And the third one is that they use on this dissimilarity measure, they use a hierarchical clustering, and they cut the tree in some way, and cutting the tree gives you the different groups. Okay, so this is in very general the approach of WGCNA that we also used. Okay, so when you use it, you get different modules. I must say that when you use it in the whole brain, the resolution is very, very low, because the brain composed of so many different cell types. There is a very strong correlation, which is really an effect of the composition of the cell types in each part of the brain. So many of those modules that we get actually capture a specific cell type, like specific type of neurons, or astroglia, or microglia, etc., etc. Okay, so the resolution is very, very low. And we've tested the enrichment in each one of those modules for common and rare variation. And we did get two modules. Here in red means higher enrichment, green means lower enrichment. So we got two modules that were significantly enriched for both common and rare variation. At the time, we were very happy because we were not sure that common variation really contribute much to autism. And doesn't matter the details, but one of them included synaptic genes, and those type of genes were more enriched with common variants, as you can see here. And another group, which included many transcription regulators that are expressed in specific brain regions that are involved in neurogenesis, generation of neurons. And this group of genes were more affected by rare variants. So this analysis at the time, this is the best we could have done, but also because of the data that we had. But there were many problems with this analysis. First of all, we studied adult brain while we were looking for genes that contribute to neurodevelopmental disorders. Where's the development here? Another thing is that we had a list of rare variants that were identified by a very biased search. People would get in love with a gene, sequence it, find some mutations, and associate it with the disease. And also there are all kind of biases, for example, in mutation rates that could bias your analysis. One known problem in this type of analysis is that is the size of the genes. Many of the brain specific genes are very, very large. So you need to take this into account. So with new technologies, we had other data to work with. We've studied mutations, de novo mutations from unbiased screens from exome sequencing studies, looking at single nucleotide variants. And we also had the release of gene expression from the Allen Brain Atlas on developing brain that we can use to analyze it. And here we've looked at different types of point mutations. We've compared the wild type to loss of function or disruptive mutation, or also describe those names. Talking about mutations that in many cases produce a stop codon and are highly likely to be functional or disruptive. Compare them to missense mutations that could be functional. Maybe they are, maybe they are not. Here again, we have almost no real good methods to assign a prediction about which mutations, the missense mutations, are functional and which one are not. And we also used the, was talking about silent or synonymous mutations as our control, because we assume that probably most of them are not functional. Okay? So we've compared those different types of mutations. And one of the things that we did was to look and see whether mutations, those de novo mutations, affect genes that are active during development or during other periods. Okay, so we've actually divided all the genes in the genomes in the ones that are expressed in the developing brain and the ones which are expressed in a different pattern, and compare the different type of mutations. And this is what you get, is that the de novo mutations are preferentially disrupt genes active before birth. And what we did here was to compare the rate of disruptive mutations here in blue to silent and missense mutations. Okay? And you can see that the rate here for disruptive mutations is much higher in autism in developmental genes versus other genes in the genome. Okay? Similarly, we could look at pathways in those genes, and when you do this kind of analysis, you see very clearly that there is a strong enrichment for genes that are active that are active in chromatin organization, which is very, very surprising, because chromatin, it's a very general thing. It's important for every cell that we have in our body. Chromatin is involved in the packaging of the DNA, regulation of expression, cell cycle. It's a very, very general thing, and why should chromatin be involved in autism? It doesn't make sense to me. So one of the questions was, how specific is this type of enrichment to autism, or whether this is something more general. As I said, why should chromatin be associated with autism? Something so basic in cell function. The other thing is that there are a strong genetic overlap between different neurodevelopmental disorders that we know. So also hint into the possibility that this is not very specific. So some of those chromatin regulators are mutated also in other disorders. So for example, if you look at autism, you have a very large list of genes that are mutated, very large list, and each one of them we have only very few samples, very few patients, but each one of those genes, just looking at them, you find that they are associated with many different neurodevelopmental disorders. I'm just showing you here just an example of one such a gene. Now, we have many problems. I mean, the fact that we see some overlap, what does it mean? So, one problem that we have, we see those de novo mutations also in control individuals, also in healthy subjects. In many studies, they don't have controls at all. Because they've collected the samples years ago, thinking about association or transmission disequilibrium tests, and so they have only 2 parents and a child. Even if you have controls, those de novo mutations are very, very rare. So in each genome, you're going to have very few of them. So the sample size is very, very small. So there have been several solutions to this problem in the field. One of them is to simulate random mutations. So this is, for example, the DNA-enriched software. You can give it a list of mutations, specific type of mutations that you observe, and it will randomly simulate mutations for you. So this is instead of having a control sample. Okay. Another thing that was before. So you can use, for example, this type of program to show that there is a significant overlap between disorders. And it's not just that it looks like there is an overlap, but maybe I'll skip it. This is just to show that there is a significant overlap. Another solution to this problem is based on the huge exome sequencing that happens everywhere and people been aggregating all this data, and people from Mark Daly group have calculated for each gene in the genome how intolerant a gene is to mutations, and this is something also that you can use in those studies. Okay, so for each gene in the genome, we have a constraint score that tells you how intolerant the gene is to mutations. And this framework, it was published in Nature Genetics. Actually, what you have is a Z-score for each genes that compares the expected number of mutations to observed number of mutations. And if you look at this score, what you can show, they also found it, is that the genes that are mutated in neurodevelopmental disorders are also under a very strong selective constraint. So here in this graph, which I'll just briefly show you, is here in the y-axis is this Z-score calculated in this framework and this is the average of this score for genes with different mutations across different neurodevelopmental disorders. And as I said, we see an association between being intolerant to mutation and being a gene mutated in neurodevelopmental disorders. And very surprisingly, if you look at those genes, let's say you take the 1,000 genes, the most intolerant to mutations in the genome, they code for genes which are important specifically to the brain, not to the lungs, not to the kidney. So if you look at where those genes are expressed, they are specifically expressed in the brain. This is one type of this kind of analysis that I'm not going to go into the details, but this is a method that was developed by Dugarty lab. So you have specific enrichment of those genes intolerant to mutations in the brain. Those genes are specifically involved in generation of neurons, neurogenesis. Many of them are chromatin regulators. Okay, so those chromatin regulators are not specific to autism, but tells you something about being sensitive to mutations. I don't think I have time too much.
5 minutes, yeah.
There's no, you are the chair?
Yeah.
Ah, okay. I thought everybody's his own chair. This is what I understood from the course.
But you can question authority
Huh?
if you want.
No. Okay, I'll just want to finish with 2 minutes and tell you that not only that those chromatin regulators are and those genes are not associated specifically with autism, the specific pathways that we see really depends on the type of mutations that we look at. Work done by Shochat. I don't have time for questions. Okay.
Is there any evidence, so you see, many of these genes seem to be active in the brain. Did people find any pattern in terms of when they're active? Because a little
Yes.
earlier we were speaking of...
So the question was when those genes are active. I will show you.
Okay.
So it really depends on the type of mutations that you're looking at, okay? So if you look at de novo mutations and you separate them to loss of functions mutations and missense mutations, you actually get different type of genes from pathways, and they're expressed in different time points in the developing of the brain. So if you look at loss of function mutations, we see enrichment for chromatin and transcription regulators that are expressed very early in development. They are involved in proliferation and differentiation, early proliferation and differentiation. If you look at a group of missense mutations, we see different pathways, such as pathways that or genes that are involved in more later stages of the development of the neurons. While if you take mutations that are recessive in individuals with, for example, autism or intellectual disability, completely different pathways. This will be mainly pathways of metabolism. Okay? So I'll just shift to the summary saying, and so if you want, I can show you later the enrichment in specific brain stages that I've just skipped. But our analysis shows that all those pathways or co-expression enrichment methods are highly biased by mutation type. So if I use one mutation type, I get one result. If I use a different mutation type, I get a very different type of pathways. I think that when we analyze those mutations, what we reveal are not the processes that cause a disease, but processes that are sensitive to specific type of mutations. And this is a bit problematic for people who want to understand the underlying biology of disorders, how we could predict phenotypes if we don't have pathways. I don't know. So I think it's very difficult. Maybe the combination of rare variants and common variants. But because I don't have time, I'll just thank you for your attention and thank the funders. Do you want me to take any questions? Zara allows me to take if you have any questions. How many? Two.
Yeah.
Any questions?
I have one if no one answers.
Okay.
Very generic. There are many disorders, some also related to the brain, for which we have animal models, like a mouse model for
Yeah.
Is there any such thing with a lab animal for autism or some part of autism that could be used to give some more information, some more traction for some of these questions?
Okay. So the question was whether there are animal or cellular models that we can use to answer those questions. And so the answer is yes. As I've showed you in the beginning, many of the things we do in my lab is actually looking at mouse and cellular model. And it's quite surprisingly, when you take those chromatin regulators, or the ones that we've been looking at, and you mutate them, you get in cells and in mice phenotypes that are highly related to what we see in the humans.
In autism.
So I want to say something, okay, to be clear.
Yes.
There is no gene for autism. There's nothing like that. There are genes that are involved in development of the brain, and if you have a
Is there a strain of mice that
Okay.
reproduces autism?
I'll explain myself. Okay, because there is no gene for autism. There are genes that are involved in the developing of the brain, and when you mutate those genes, in some individuals, you're going to have behaviors that we will diagnose as autism. This is what we have. We can take those genes and make mouse models with mutation in those genes, and we can look at the behavior of the mouse. In many cases, we're going to see social deficits in the mouse that may copy in some way the condition in humans, but it's not going to be an autistic mice.
Yeah, but it's a proxy for it, the same way you have okay.
Okay? Thank you.